got to do that. Okay. So the urinary system. So as we cover that, why do we have it? We've touched on it a little bit in the past, especially in the introduction slides. I want to go in a little bit more detail. So basically, this consists of, I got it to work. I plugged in the HDMI cable and it started. Interesting. I've never had that happen quickly. Yep, I didn't expect to do that, but it took off the transition and automatically reset. So
Very
something you remember in the future.
okay, great.
Thank you. I'm sorry to have you walk. Okay, getting back. So this consists of the kidneys, the ureter, the bladder, and the urethra. Those are all aspects of your urinary system. And basically, why do we have it? Well, it has a number of functions. One of those is to filter stuff, which we'll spend a fair amount of time talking about today. But one of those things is water, and this maintains water balance in your body. This also, in respect to your circulatory system, is maintaining blood volume. It also is critical for regulating various ions and other molecules that are in plasma, such as salts and sugars and amino acids, etc., bicarbonates, acid ions, etc. All right. Now, because it can control hydrogen ions as well as bicarbonate, it's going to be critical in regulating blood pH. All right. So, because remember, blood pH is neutral at 7.2 to 7.4. It needs to be maintained. The kidney plays a critical role. Now, also, this is critical for getting rid of metabolic wastes, as well as toxins and poisons and so on. All right. Also, remember, as we've already covered in the circulatory system, in terms of red blood cells, the kidney is also an endocrine organ. It secretes erythropoietin, which is critical for red RBC differentiation from hematopoietic stem cells in the bone marrow. It also is important for another hormone called renin, which we'll talk about later today. All right. All we have already also covered how it plays a role in the activation of vitamin D3 to its active metabolitic form. All right. So all of these are functions of the urinary system. This is a cross section of one of your kidneys, right? So you'll have two major vessels moving into that. You'll have your renal artery, and then you'll have your renal vein. You also have your ureter, right? This here then is draining what does not get reabsorbed or goes back into the bloodstream. Ultimately, once it enters the ureter, we call it urine because it's going to go to the bladder and ultimately be eliminated as urine, all right? Now, one thing that we have, note the loop-type structures of your vasculature. These actually form on what we call these three-dimensional pyramids, if you will. And they play a critical role because this is where the actual nephrons are, and they do the primary work of the kidney. We'll get there momentarily. The kidney is totally encased in something called a renal capsule. This is a fibrous membrane which delineates the kidney itself. All right. Now, there is the ureter is accepting stuff from the calyx. So think about the major calyx as kind of like a funnel. And so thing goes, you have stuff goes into collecting ducts, eventually the minor calyx, the larger calyx, and ultimately out the ureter. All right. So this is your kidney. Now, let's focus on that smallest functional aspect of the kidney. We call it a nephron. So you have hundreds of nephrons in your kidneys across the two of them. And each of these contains a vascular component and a tubular component, right? Now, basically, these are responsible for doing the primary functions of the kidney unassociated with, endocrine functions and so on. And that is they filter plasma. But that isn't all they do. They also play a critical role in reabsorption and secretion. These are the three primary functions of the nephron, right? This is to differentiate it from, say, a dialysis machine or an artificial kidney. It only does one. All it can do is filter plasma. It cannot elicit or engage in reabsorption or secretion. So if you get hooked up to a dialysis machine, it will filter out by size exclusion, water, small molecules, and wastes and so forth, but it doesn't return any of that. So that means you have to physically ingest it by consuming more liquids, replacing those salts and sugars and things that are lost. All right. But it is tremendous the way the kidney can function and this breaks down to the smallest functional component, which is the nephron. Now, because we have this combination of filtration, reabsorption, and secretion, this enables the regulatory aspects of the kidney in terms of water volume, pH, osmolarity, etc. All right. Now, and again, like I mentioned before, a nephron consists of both a vascular component as well as a tubular component. Now, the vascular component is referred to as the glomerulus, and then there is an afferent arteriole leading to this glomerulus and an efferent arteriole that drains that glomerulus. The tubular component begins as a structure that surrounds the glomerulus called the Bowman's capsule. This begins the tubule. That then drains into something called the proximal convoluted tubule. From there to the loop of Henle. From there to the distal convoluted tubule. From there to the collecting duct. From the collecting duct into the calyx complex. And ultimately, whatever's still in that lumen is ultimately going to be expelled as urine. All right? So these are the functions. I'll show you a picture. Here is a simplified picture of the anatomy of a nephron. So let's start with the vascular component over here. Here's the red vessel, and that's going to denote, in this particular case, the arterial side. All right, so we have an arterial side. We begin with the afferent arteriole. It then proceeds to the glomerulus, and that glomerulus there looks something like this. I'll use my hand here. So this is the afferent arteriole here. Here is the glomerulus. Here's the efferent arteriole. So blood enters here. It's filtered through the glomerulus, and what doesn't get filtered then exits via the efferent arteriole. From there, it goes into the peritubular capillaries, which you see here, ultimately leaving the nephron via these venules over here. All right. Now, let's focus on that glomerulus. Surrounding that glomerulus is this funnel type structure called the Bowman's capsule, which is the beginning of the tubule. All right. So basically, anything that is filtered across that glomerular membrane is plasma, because that's the liquid part of blood. But once it gets filtered, it is no longer plasma because it doesn't have the same constituents. It's referred to as the tubular filtrate. This is captured by the Bowman's capsule, this funnel device. All right. Now it's called the tubular filtrate. Now, it will then proceed to the proximal convoluted tubule, the loop of Henle, which consists of a descending and an ascending loop, then the distal convoluted tubule, and then ultimately draining into the collecting ducts. All right. Multiple nephrons drain into a collecting duct. Now, look up here at where the glomerulus and the Bowman's capsule are. You see that little greenish shaded area? This refers to the juxtaglomerular apparatus. I repeat, the juxtaglomerular apparatus. It consists of the efferent and the afferent arteriole, the associated glomerulus, and a structure that goes up like here, which represents the distal convoluted tubule. All right. Note that the tubule, here we go in the loop of Henle, we're going up into the distal convoluted tubule, and there it goes up between the afferent and the efferent arteriole, and those structures right there, primarily the afferent, efferent arteriole, and the distal convoluted tubule, that section there represents the juxtamedullary apparatus. Okay, this play has a regulatory role I'll get into in a little bit. I'm just introducing the anatomic pictures right now. All right. Now, the tubule consists of cells that line this whole tubule, and they're called renal tubular epithelial cells. Now, even though they're basically epithelial cells, they line this entire length of this tubule. As you go along this tubule and you're in different parts of the tubule, those cells have different function and they look differently. Okay, they're all epithelial cells, but they look different. Okay, that's because they're doing different functions depending on where I'm at along the length of that tubule. If I'm in the afferent arteriole, I will do different things than in the descending loop of Henle, different from the ascending loop of Henle, etc. All right, but they are still basically the same types of cells. Now, there are basically two types of nephrons. We have them called the cortical nephron, and then we have something called the juxtamedullary nephron. The distinction depends on where they exist within the kidney. So the kidney, just like the adrenal gland, is divided into an outer cortex and an inner medulla region. All right. In the kidney, there is where your cortex meets your medulla. This is called the juxtamedullary interface. Okay. So hence if we have nephrons that exist on this zone area, which has significant constituents both in the cortical and the medullary space, it's called a juxtamedullary nephron because it's right there at the interface. Then there are nephrons which exist almost exclusively within the cortical aspect, and these are referred to as cortical nephrons. So we have cortical nephrons, juxtamedullary nephrons, and it all depends on where they are in the kidney. Are they primarily in the cortical aspect, or are they at the cortical medullary interface, right? Now, cortical nephrons in humans are the most numerous, and basically, as I've already mentioned, they're almost exclusively within the cortex. Some small region could extend into the medullary space. Juxtamedullary nephrons exist at the interface of the cortex and medulla. They have a significant portion of them that extends deep into the medullary space. That is the loop of Henle for number one. And then there is an associated modification of an efferent arteriole called the vasa recta. And this loop of capillary also extends deep down along in parallel with the loop of Henle. I'll show you pictures later. All right. So these are the two basic types of nephrons. This is again showing you, here's cortical nephrons, only a small aspect extends down here into the medullary region, which is a shaded region. Over here, we have a juxtamedullary nephron. You have the loop of Henle extending deep into the medullary space, and we have this vasa recta also extending in this space, right? I also want to just take a moment to remind you the kidneys are endocrine organs, and they're also involved in gluconeogenesis. About 1/5 of gluconeogenesis during fasting occurs in the kidney because all of the transport functions, especially during reabsorption, it requires a lot of energy. Now, let's start talking about the functionality of the nephron. Its first function is what? It's filtration. All right. So filtration occurs in only one spot, and that's in the glomerulus. The filtrate, which is once it leaves the blood, is now called tubular filtrate. This is captured by the Bowman's capsule, and then it drains into the tubule, and then we can have reabsorption or secretion. All right. Now, reabsorption and secretion, you only have filtration at the glomerulus. You have reabsorption and secretion. Depending on where you're at, things are different along the length of that tubule. All right. In the proximal convoluted tubules, you can have both reabsorption and secretion. Notice the arrows. If the arrows are leaving or pointing away from the tubule, that's reabsorption. If they are pointing to the tubule, that's secretion. Right. Now, secretion is completely independent of filtration. all right? Just as reabsorption is. And basically reabsorption is of the tubular filtrate. Secretion is adding stuff to the tubular filtrate, right? Now, the loop of Henle is unique. The descending arm of the loop of Henle is only, I repeat, only permeable to water, not salt. Only water. And that's via osmosis. Now, the ascending loop is impermeable to water, but permeable to salt. It is both passive and active transport of salt occurs along that ascending loop of Henle. When we get to the distal convoluted tubule, we have both reabsorption and secretion. And then we can get into the collecting duct, and there's some additional reabsorption and reabsorption can occur as well, right? So these are all the things that are happening in the nephron. So if we start with filtration and yes. The ascending loop of Henle is impermeable to water, not permeable to water, permeable to salts. Salts can be transferred, basically sodium, can be transferred via both passive and active transport. All right, now let's focus on filtration. Now, I want to do this because what happens in the glomerulus is unique to the glomerulus. It's unique to the nephron. It's unique to the kidney. It's the only place it happens, right? So basically, filtration within the aspect of the glomerulus is a function of three things. It's a function of the total surface area available for filtration. Remember your fix. It is the permeability of that membrane, and it also is dependent upon the net filtration pressure. Now, the unique nature of the glomerulus forms this network of multiple little capillaries that have an entry and a drainage. All right. Now, that means that it's increasing surface area for filtration. We will talk about the unique nature of it as a filtration membrane in a moment. It is the most efficient filtration membrane in the body. All right. And we'll get there. Now, what also is important is the net filtration pressure. We'll discuss where that comes from. But again, it is unique to the glomerulus. And basically, the filtration membrane itself is thousands of times more permeable to water and solids than any other comparable capillary membrane, which we've already covered in prior. And the glomerular pressure is higher than other capillary beds. And this is a function of the unique anatomic structure of, here again, the glomerulus here, afferent arterial, efferent arterial. The reason I'm showing you this is there's a big size difference between the afferent arterial and the efferent arterial. The afferent arterial is a bigger vessel, i.e. it has a larger radial luminal area than the efferent. What does that mean? That means that there's a pressure buildup here. Okay? So if there's a size difference going in, it's a bigger hole and then it's a smaller hole going out. So that means you're maintaining a higher pressure across the glomerulus. In fact, it's 55 millimeters of mercury. All right? Now, filtration is obviously size dependent. If it's small enough, it fits through the holes, it goes through. These are less than 3 nanometers. Then as we get a moderate size, there is less efficient movement. And then, of course, if we get too big, it can't go across. All right. It's a standard size exclusion filtration membrane. All right. Now, what I want you to note here is that we have the renal artery. The pressure is very high because that's very close to your heart, and the blood's coming down. And then, of course, the glomerular capillary is the pressure stays high. This is to help facilitate filtration. And again, this is due to the anatomic differences in the size between the afferent and the efferent arterial. Right? Now, again, I've already mentioned it's non-selective, and basically 1/5 of the plasma that is going through the kidneys gets filtered. via the glomeruli, all of those glomeruli in each of those nephrons. And so there's a couple of things I mentioned three things: surface area, all right, and I mentioned the filtration membrane permeability, and I mentioned pressure. We do not, on a day-to-day basis, we cannot affect the surface area. That's fixed, as is the membrane filtration permeability. The only thing that can be varied moment to moment is pressure. Okay, that's the net filtration pressure. The net filtration pressure is a function of the outward forces, which is basically blood pressure, that's at 55 millimeters of mercury, and then resistive forces, which we will talk about in a moment. The net effect is we are maintaining the efficiency of this filtration system is such that in a 24-hour period, 180, almost 200 liters of plasma gets filtered out of the blood and becomes tubular filtrate in a 24-hour. Think about that. Essentially 200 liters of plasma a day gets filtered by both your kidneys. Now, obviously, we're not urinating that quantity of urine. We're only, let's round it to 1 liter a day. Okay, so probably a little high, but keep the math easy, right? So if we go from 180 or 200 liters a day, that means we're over 95% efficient in reabsorption of that water because it is not leaving us as urine, right? So that only happens because of reabsorption. Now, when you think about that, that's an extremely efficient filtration and reabsorption process, right? Now, net filtration pressure is a function of three variables. It's a function of the pressure within the blood pressure in the glomerulus, all right? And that is 55 millimeters of mercury. This is enabled because the afferent arteriole is much larger in diameter than the efferent. That creates that pressure. How many of you know what a garden hose is, right? Especially if you get those little collapsible ones and you turn on the water, all right? Now, just the end of the hose, don't worry about a spigot because you can press those hoses, right? So you turn the water on, it's just a little bit coming out, right? As you press, what are you doing? You're closing that orifice, right? So that means you're creating a back pressure back here. What's going to happen is instead of the water going like this, it'll be spraying out there. And that's because you have a pressure buildup behind that partial clogging. That's what's happening here. I have more plasma entering the glomerulus than leaving. So that induces a higher pressure, which facilitates that's a pressure moving water out of plasma, out of the blood. Now, there are two resistive forces. One of those is the blood the blood colloid osmotic pressure. I've already talked about that in bulk flow, right? This is a capillary. It happens here too. These are the proteins that do not normally get filtered. In other words, normally they stay in the blood and they want to hold on to water, right? And so this is resisting water leaving blood via the glomerulus, and this is referred to, and this is around 30 millimeters of mercury. The additional resistive force is the pressure in the tubular filtrate. There's that pressure, that back pressure. So it isn't just going into emptiness. There's already fluid in there, but it is minimal. It is only around 15 millimeters of mercury. So the total resistive forces are 30 plus 15 or 45 millimeters of mercury. We subtract that from the outward force of 55 millimeters of mercury. We end up with a net filtration pressure of 10 millimeters of mercury. It doesn't sound like a lot, but that's enough to move essentially 200 liters of filtrate a day for you and your kidneys, right? Now, I'm going to allude to this since I have this picture up. I'm going to mention it here. How can I change this? I can't change this glomerulus itself. I can't change the filtration aspects of that glomerulus. The only thing I can change is pressure. And the way I change that pressure is I influence the pressure, outward pressure here. So if I change the diameter of that afferent arteriole, I can change pressure. What happens, let me ask you, what happens if I reduce the size of that afferent arteriole? That means it is approaching the size of the efferent arteriole. Will I increase or decrease pressure across the glomerulus? Decrease, correct. And if I further increase, I will have the opposite effect. Okay? It's that simple. So day to day, moment to moment, your kidneys regulate the net filtration pressure by changing the size of these primarily the afferent arteriole, but you can also change it by efferent. What happens if you have a global stimulation of the sympathetic nervous system inducing extensive vasoconstriction? You can close off the whole thing, both afferent and efferent arterioles. Okay. All right. So that gets us to the intrinsic controls. I'll mention it here and go into more detail in a little bit later. But intrinsic controls, as you remember from prior lectures, that means everything occurs in the kidney, within that tissue. Extrinsic controls means some element has to be outside the kidney. So intrinsic controls, there are two aspects. There's the myogenic mechanism, which means that if you have an elastic tube and you start to put increased pressure inside, what's going to happen? You're going to get the elastic starts to stretch, that rubber band, and wants to return back to its normal resting position. So as you increase the size of the afferent arteriole via increase in pressure, it's going to want to contract back. Okay, that is a negative feedback. So that is the first intrinsic control. The second one is called the tubulo-glomerular feedback mechanism. I'll explain that in just a bit. All right. Then, of course, sympathetic nervous system, since that's the brain, that's also an extrinsic control. Right? Now, let's go back and then, of course, how is that regulated? That's going to be regulated by the juxtaglomerular apparatus, because I mentioned here is your glomerulus, your afferent arteriole, and here is your distal convoluted tubule. This juxtamedullary nephron consists of things that are in the distal convoluted tubule, which are measuring the filtrate. Remember, this is just before it's leaving the tubule. So just like in a standard chemical engineering processing plant, you are measuring stuff all along the process, and that provides feedback. So the same thing here. So that juxtaglomerular apparatus has groups of cells which are measuring the filtrate. They're measuring osmolarity, etc., and they provide feedback. We'll get to how that works momentarily, but I'm just expressing that's where it happens. All right. Now, let's go back to this filtration membrane for a moment. It's extremely efficient. However, and we've talked about different types of capillaries. We've talked about capillaries in the brain. We've talked about capillaries in most tissues. Now let's talk about capillaries that represent the glomerulus. All right. Now, so basically, filtration is size exclusion. So that means there has to be some holes. So there's two types of holes that are forming this glomerular filtration matrix. And then the first one are the endothelial cells. So the endothelial cells have fenestrations. Remember, I touched base on that. Those are little holes. And then there are additional cell structures that surround the glomerular structure, and they're called podocytes. And these form secondary size exclusion vehicles. And I'll show you a picture in just a moment. All right, so let's go up here. There's two things I want you to see here. Let's start inside the Bowman's capsule. There is your glomerulus, and then the red structure is the glomerulus, and then the white structure with the purple nuclei, those are called podocytes, right? So podocytes, let's drop down here. So here's the endothelium, the capillary. Notice the fenestrations of the holes. So that means anything can pass through those holes if it's small enough. All right. Then around this, we have the podocytes, and you have these these are filtration slits that are formed between adjacent podocytes. And so stuff that's being filtered from the blood has to go through these fenestrations and then through these slits. All right. Now let's go up here to the top and look over here to the right of the top panel. There you see the afferent and the efferent arteriole. Afferent is the larger vessel. Efferent is the smaller vessel. And then you have this is the collecting duct, which is coming out this way. Here are cells in there called macula densa cells, and they are measuring that tubular filtrate, qualities of the tubular filtrate, everything from osmolarity and so on. And there's additional cells called, there are juxtaglomerulary cells, and there's mesanglial cells, all right? And so they are involved in this juxtamedullary apparatus. And that does two things. It's capable of both intrinsic and extrinsic controls. We'll get to the details later. I'm just showing you the anatomy here. Let's focus on that filtration membrane, the glomerulus. So here's blood on the left side of this panel. It has to go through a fenestration hole. Then this little white part here is basically extracellular matrix, which forms a basic tube that the endothelial cells bind to. But there's no, basically, the holes, there's no defined porosity. So it has no size exclusivity to it. So things that are being filtered have to go through the fenestration, and then it has to go through the podocytic slits. Note that the space between the podocytic slits is smaller than the fenestrations. That only makes sense. So there's a two-level filtration. It has to go through both these sets of holes. Now, next is this. Filtration is extremely efficient. Think about that, 200 bloody liters a day through those two little kidneys about the size of my fist. Now, all of that's happening across the glomerulus. So, filtration, blood flow is in a single direction, all right? The holes are transverse to the flow of blood. How many of you have used filtration membranes? Any of you? Okay. So think most filtration membranes are vertical, in line. You have the filter and the flow is going this way. All right. So what that means is those holes are subject to being clogged. And it happens all the time. All right. You put complex medias in there, blood, whatever, and if things are big and whole, they eventually will clog the holes. It's not particularly efficient, but depending on what you're trying to do, it works well. All right. But if you want to do large volumes, no, it is not scalable. That gets us to the way the kidney works. Note that the blood flow is this way. The holes are transverse. All right. And so that gets us to a filtration system, which is called TFF, transverse filtration. And what that means is just exactly what's happening here in the glomerulus. I can do size-exclusive filtration instead of going vertical to the hole, transverse to the hole. And what happens is this process is scalable because they're not subject to clogging. Okay? So the big particles are always in a flow field going this way, and the small stuff comes out, the big stuff stays here, but it doesn't tend to clog. Okay? And TFF is used. It's almost infinitely scalable, extremely efficient, comparatively speaking, to a vertical concentration, right? Okay, we have filtration. We now have a tubular filtrate. We now need to get the bulk of it back into the blood, all right? That's referred to as reabsorption. Now, what gets reabsorbed? All the critical things, water, salts, sugars, amino acids, etc. These bicarbonate and so on, these need to be reabsorbed. They're extremely expensive to get into your body in the first place. So basically, we need to recover them. Now, reabsorption is distinct from filtration because it's specific. Okay, it's specific. Filtration is only specific due to size, but it's non-specific otherwise. Filtration is specific for the particular molecule we're talking about. First off, remember, water is always transported via osmosis. It follows, it's going down its concentration gradient. So think of it, it follows salt. All right. You move the salt, water will follow. Right? Fine. How do you get salt across? You pump it back across. Active transport. We've already talked about that. So you have preferential active transport not only for salt, but for glucose, amino acids, and so on. All right. Now, the next thing we need to talk about is this specificity. And not only is it critical, you can have differential specificity, and the amount of reabsorption varies depending on, do I need to recover? Do I need to recover more sodium? Do I need to recover less potassium? Do I need to recover more sugar? Do I need to recover less sugar, etc.? But it's very specific, right? Now, where does it occur? Just as I mentioned, primary, it can occur in the proximal, distal convoluted tubule as well, depending water in the descending loop of Henle, salt in the ascending loop of Henle. So depending where you're at in the tubule, different things can be reabsorbed. All right. Now, reabsorption can occur if things can go between these tubular epithelial cells, or they have to go through. And the rules that we've covered throughout the introduction and throughout this are apply. All right. So the thing to remember is this: filtration is based on size. Everything, osmolarity is what? Let's exclude sugars. It's basically salt and water. Okay. Then your blood has an osmolarity. Now that means it has a salt concentration that is say 300 milliosmoles. Right? Now, filtration that means everything that contributes to osmolarity gets transferred during filtration. So there is no initial difference in the osmolarity, the salt concentration of what's in plasma and what's in the tubular filtrate. All right? They're both 300 milliosmoles. But what happens is, so that means that if I want to move salt, if I want to recover salt via reabsorption, I can't go down a concentration differential because there isn't one. It's the same on both sides. So the only way I can get reabsorption is by active transport. So I have to actively transport that salt, the amino acids, those sugars, right? Now, the next thing to remember is this. Reabsorption, filtration is going from blood across the glomerular membrane into the tubular filtrate, all right? And goes right into the tubule lumen. Reabsorption is more complex. It goes from the tubular lumen across the epithelial layer, lining of the tubule into the interstitial compartment, and from there into blood. So there always is this intermediary area, the interstitial zone, of which anything being reabsorbed or secreted has to cross, right? So we have reabsorption. It goes first into the interstitial space, and then it has to go into the capillary, the peritubular capillaries. Now, these are not near as complex, all right? So basically, they will go into here down their respective concentration gradients. The only active transport is primarily here, all right? Secretion is also specific, and that occurs as well as reabsorption. And it's primarily used to control things like hydrogen ions, which is going to help you regulate the pH of blood. Also, it can be important for getting rid of excess potassium. Remember your membrane potential. There's more potassium inside the cells than outside the cells. There's more sodium outside than inside. If you have a huge level of potassium increasing, that's going to have a negative impact on your membrane potential. So the body needs to regulate those interstitial concentrations of potassium. So if they get too high and I can't get rid of, they're so high I can't get rid of them by simple filtration, I have to have additional secretion of that potassium in order to drive those levels down. All right. Okay. there are about 4 slides following one another, and they're talking about something very specific. And they're talking about the interaction between the loop of Henle and the vasa recta. Remember, these are part of the juxtamedullary capillary, juxtamedullary nephron. Now, the primary role of this is reabsorption of water, primarily reabsorption of water. All right. So what I want to do is talk how this functions. Now, this tends to be complicated. Normally, years ago, I used to only show one slide, but a lot of you will have trouble with this concept. So I've increased the number of slides over the years. They're all basically saying the same thing, but I'm hoping that together they are more helpful. All right. So what you're looking at here, let me define what you're looking at. Here is your loop of Henle, right? This is a collecting duct, and here's your vasa recta. Remember, the vasa recta is a modified efferent arteriole. Now, they're like this, but the way they actually work is this vasa recta is laying like this. So they're next to each other, right? Also, let me clarify that blood flow direction and tubular fluid flow direction are the same. They're moving in the same direction, and they're parallel to one another. All right. The next thing I want you to look at is the osmolarities within the medullary space. So remember, here's your cortex from here up. Down here is the medullary space. As you go toward deeper into medullary space, the osmolarity of the interstitial fluid goes up. All right. It starts out at 300 milliosmoles, which is equivalent to blood. As we go down to the deepest aspects of the loop of Henle and the vasa recta, it's at 1200 milliosmoles. All right. So there's a gradient. There's an osmotic gradient going from 300 to 1200 milliosmoles as you get deeper into the medullary space. Why is this important? It's important because remember, how does water go? It goes down its concentration gradient. So wherever the concentration is less, you're going to have water go toward the less concentration, right? Now, so at initial filtration, remember, I've already said that the osmolarity between plasma and your initial tubular filtrate is the same, right? So that means that you need to, remember, the descending loop of Henle is permeable to water. But what's going to influence water leaving that tube is that it needs to go down its concentration gradient. It's at 300. What happens as it gets over here and maybe it's up to around 400? So that means there's less water on the outside. That means that water is going to leave in order to equilibrate. As we go down, it goes all the way up to 1200 here. So water's leaving. But remember, it's reaching equilibrium. So depending on where you're at, you start at 300 here. As you get to the bottom, it's 1200 because it's balancing out. The water is leaving, but it's not permeable to salt. So only water can cross. So water is leaving the tubule, the descending loop of tubule, at such a rate, so it's maintaining the relative osmolarity at in the tubular side versus the interstitial side. And it goes from 300 all the way up to 1200, where that water goes into the interstitial compartment. Yes? I'll get to that. Okay? Right now, I just want you to be aware of this gradient of osmolarity, all right? And how that's pulling water out of the descending loop of Henle. And the water is going here in the interstitial space. All right? Now, we get to the ascending loop of Henle, which is now impermeable to water, but permeable to salt. So its goal is to bring salt back in. All right? So salt comes back in, or salt goes back out, rather. And that means that additional salt is going out. So note that it goes from 1200 all the way up to almost 100 here. So that means it's pumping salt back out, or pumping salt out. So water is leaving during the descending part, and then this And then there's a lot of salt left in the tubule, but before that filtrate leaves that ascending loop of Henle, it's lost most of its salt. It can even be below the osmolarity of blood. In this case, it's up down to 100. All right. There can be additional reabsorption and secretion occurring in the distal convoluted tubule as well as in the collecting duct. But let's just focus here. Now, that's the tubule. Water's leaving in the ascending loop and the descending loop of Henle. Salt is leaving in the ascending loop of Henle. Everybody with me so far? Now, it goes in the interstitial space. Also, remember, there's essentially 200 liters of water being filtered every day. Right? So, wait a minute. Where's the water going? All of that water goes, think about all that water. If it goes to the kidney and doesn't go someplace else, what's going to happen? Your kidneys are going to explode. So the water's got to go someplace. So the water goes back here into the blood. All right? Because remember, this is beside this. So water is going into the interstitial space, and then correspondingly, it's going down, returning to blood down its concentration gradient. And we can also have some recovery of salt or not, depending on where we're at. So remember, this is laying over here. So in this descending part of the loop of Henle, that corresponds to the descending loop of the vasa recta. So note that during that, we have an increased salt concentration. So salt is going into here, and water is leaving because it's normally at 300 milliosmoles, just like over here. However, in the ascending loop, then what happens, because now salt is going to be leaving and then water is going back. And so water is getting returned during the ascending part. All right. So this is what's happening with the juxtamedullary nephron in respect to the vasa recta and that. And the vasa recta is responsible for, we have to have this gradient. But if all this water gets over here, that's going to dilute our gradient. It goes away. And we have to have the vasa recta to take out the extra water in order to maintain that gradient. So it's responsible for maintaining that concentration gradient. Right? We call this the counter current. Now, some students mix this up thinking that blood flow direction and tubular flow direction then must be different. They're not. They're in the same direction. That is not what countercurrent refers to. Countercurrent refers to this simply. The descending loop of Henle is permeable to water. The ascending loop of Henle is impermeable to water, impermeable to salt. That's all that countercurrent means. Okay? Again, these are just showing how you establish that. So let's talk, and we call this whole complex the countercurrent multiplier. So it is determined by this unique nature of the loop of Henle. I'm not going to repeat myself. I've already done that. And then what happens is, so basically, the vasa recta forms the countercurrent exchanger because this is responsible for maintaining that gradient within the interstitial compartment, which is pulling the water out of the descending loop of Henle. All right, so this is the countercurrent multiplier. So do be aware of this for upcoming exam in about a week and a half. So, this is just showing you again active transport and so forth, and it's bringing in the collecting duct. To this point, I've kind of ignored its functionality, but it also plays a critical role. Now, endocrinology part. Can't get away from those hormones. All right. Now, these hormones, some of them I have discussed, introduced to you in earlier places, but I'm going to, and spatially in the, I touch base in terms of the circulatory system. I talked about some hormones, but I didn't really talk about what they were. I want to talk about what they were, what they are, and what they do in respect to the urinary system. So let's begin with the adrenal cortex and a hormone called aldosterone. All right. Remember, it's critically important to maintain the right ratio of sodium and potassium across the cell plasma membrane. You want to have more potassium inside, more sodium outside. What happens if that flips? We need to get rid of the extra, all right? Or, so let's say that your blood, so what happens, your blood, you have too much potassium. We need to get rid of that extra potassium, more than we can simply filter out and not reabsorb, all right? So that means secretion, all right, or lack of reabsorption, right? So basically, this is sensed in the adrenal cortex. These are basically, we're looking at either a high potassium or a low sodium level. As a response, you will have the release of aldosterone. It's a steroid-based hormone, just like cortisol or the sex hormones. And this will then target the renal tubules, where it will direct two opposing things. Number one, it will increase secretion of potassium and increase reabsorption of sodium. And that changes the ratio back to what it should be. All right. So that's the purpose of aldosterone. Now, what happens if you reabsorb more sodium? You're reabsorbing more salt. What does that mean? That means you're going to then bring more salt, reabsorb more salt too. More water via osmosis, because water follows salt. Now, the next thing, I introduced this hormone in the endocrine portion as being released from the posterior pituitary. It's ADH, antidiuretic hormone. All right. So it's produced by the hypothalamus, released from the posterior pituitary. What it does, it goes to those collecting ducts where it promotes the reabsorption of water. The purpose of that is to increase water retake, or to put more water back in the circulatory system. That's going to increase blood volume, which is going to have a direct increase on blood pressure. All right, pretty straightforward. And since we're reabsorbing more water, that means there's less water to go in urine, so we end up with a scant urine volume. All right. Okay, let's do an overview. So basically, what it boils down to, what you're looking at, we can talk about functionality of the nephron, is that the only thing we can change is net filtration pressure. So we can either increase or decrease blood pressure across the glomerulus. We can do that locally, or we can do this systemically. All right. So let's first talk about those intrinsic controls, because as we increase the pressure across the glomerulus and therefore across the afferent arteriole, it's going to want to shrink back. So that's going to have a negative impact, and that's going to reduce the size of the afferent arteriole. That will reduce pressure across the glomerulus. Next is the tubular glomerular mechanism. And the tubular glomerular mechanism involves the juxtamedullary cells. I haven't talked about how it works. Let's now talk about how it works. So it is controlled by the maculodental cells, which are integral to the distal convoluted tubule. They then measure the filtrate flow. They measure osmotic signals, and they correspondingly induce the release of vasoconstrictive hormones at the afferent arteriole. Remember, here's the distal convoluted tubule. Here is the afferent and efferent arteriole. So the maculodental cells will release components that will induce vasoconstriction of the afferent arteriole. What does that do? Vasoconstriction of the afferent arteriole means it's a smaller diameter. It's now approaching the diameter of the efferent, so that's going to reduce pressure because there's not a size difference anymore. All right? And these are both intrinsic because everything's happening in the kidney. Extrinsic, basically, that's anything that will increase overall blood pressure. Systemically, it's going to obviously increase that pressure across the glomerulus because it's going to create a higher pressure at the afferent arteriole. Everything else being equal. All right? So basically, that's what we want to talk about. So you have your, then of course, if under serious stress, maybe if we also can have stimulation, we can actually shut off flow if we have a vasoconstriction of both those arterioles. Now, let's start putting everything together. All right. So how do we control filtration? By controlling net filtration pressure. And everything we've talked about is controlling either positively or negatively net filtration pressure. All right. So basically, one of the things that you can have here is that we have our intrinsic controls, right? And our intrinsic controls. Now, our juxtamedullary, in addition to having these vasoconstrictor elements released from the macula densa cells, they also release a hormone called renin. Okay, renin then leaves the kidney. It goes into the systemic circulation where it will regulate the activation of angiotensinogen, right? So that's here, that is made in the liver. Angiotensinogen is made in the liver, and it will activate to angiotensin 1. Angiotensin 1 goes to the lungs where it becomes angiotensin 2. Angiotensin 2 has multiple effects. Number one, it will go to the adrenal cortex and promote the release of aldosterone. Aldosterone then facilitates the reabsorption of sodium. Water follows. We have an increase in blood volume, an increase in pressure, right? It also goes to induce vasoconstriction. That increases peripheral resistance, which increases blood pressure, as we've talked about in the circulatory system, right? The net effect is to have a net increase. So in the case of the aldosterone, that increases blood volume. In the case of direct effects on the vasculature, peripheral resistance, that's going to increase peripheral resistance and then leading to an increase in pressure. Also, we can have systemic arterial vasoconstriction or vasoconstriction. We also have ADH. All right, antidiuretic hormone, which means increased blood volume, increased pressure, right? So that's kind of putting it all together. Now, all of these things, basically, there is a hormone that decreases pressure as well. And that hormone is called atrial natriuretic peptide, ANP. You didn't know that the heart was an endocrine organ too. It is. It releases this hormone. And this hormone, I did introduce the hormone in the circulatory system, and I said it has global negative effect on blood pressure. So its goal is to reduce the net filtration pressure. Now, how does it work? It's global. So what it does, it suppresses aldosterone release from the adrenal cortex. It also disrupts the release of ADH release from the posterior pituitary, and it also goes over here and it reduces renin release. Because it reduces renin, that means you're not going to have the activation of angiotensinogen to angiotensin 1, angiotensin 2. In addition to activation of aldosterone, you will not have the vascular peripheral resistance increase either. Okay, the net effect is an overall global reduction in pressure. Therefore, a reduction in glomerular filtration pressure. Okay, this is putting it all together. This is everything. I'm not going to go back over it, but to remember, there you have your myogenic, which is that is an intrinsic control. You have your tubuloglomerular, which is also intrinsic controls. And then you have renin, which then stimulates an extrinsic control system because it's involving the activation of angiotensinogen. And we also have the sympathetic nervous system. And then, of course, we can bring in ANP as well, ANP, which is obviously extrinsic as well. All right. Okay. So that's it for the endocrine portion. Now, what I want to talk to you about is the role of urine as a diagnostic test. All right. Think about it. When you go to your doctor and they're going to ask you to they're going to say go to the laboratory and give blood samples. They may take a swab, and they may say pee in a cup. Okay. What I want to talk to you about, what is the value of urine? as a diagnostic because things end up in urine. And it's a completely non-invasive test. We urinate every day, okay? Under normal conditions, completely painless, right? Now, I want to give you some examples of the value of measuring stuff in the urine. So let's begin with glucose, all right? If we have an elevation in glucose, that points to what? That points to uncontrolled what? Diabetes, all right? So diabetes, let's focus on type 1 diabetes. We've known about that for thousands of years. We didn't know the cause until the last century, but we know the reason for it. I mean, basically, your body, the tissues in your body are not absorbing that glucose. It stays in the blood. We need to maintain blood glucose levels, so that means we need to filter it out of the blood via the kidneys. We don't want it back. We just want to get rid of it, so that means we're going to have extra glucose in the urine, all right? In fact, that was the first diagnostic that I'm aware of, all right? Depending on your historical documents, it's been attributed from the ancient Greeks to the Indians, Chinese, etc. So I will tell you one story that I've been told over the years, and I'll relate that one. Just, how much, I don't know. So one story is attributed to Aristotle, ancient Greece, 300 BC. He was one of the earlier, scientific aspects, not only a philosopher. So at that time in Athens, you have public toilets. And basically, he noted that some urine resulted in ants seemed to be attracted to some urine versus other urine. All right. So being the scientist, quote unquote, that he is, he asked the question, why? So he used the tools at hand, which meant his tongue. So he tasted the urine. It tasted sweet. Then, of course, you go back and you find that it's associated with diabetic patients. So that became one of the earliest diagnostic tests. In fact, taste of urine persisted across cultures all the way into the late 1800s until they developed a chemical test for sugar in urine. All right. Oh, yeah, you tasted of the urine. That was what you did. All right. Now, that's glucose. We drop on down. What about proteins? There's normally proteins in your urine, but if you have an abnormal amount of them, and especially some of these larger proteins, that denotes some pathology. All right. Maybe you're under hypertension, so you have higher blood pressure that's forcing these larger proteins across. Maybe you have some injury or something. Next is ketone bodies. That is also a sign of diabetes. Now, because you can't use glucose as energy because you don't have any insulin, what happens is you have to catabolize your other body structures in order to provide that energy. And as you do that, i.e. proteins, one of the metabolic waste products are ketone bodies. They're associated with acetone smell. If you've ever smelled acetone, that's what ketone bodies smell like. Now, there's two ways that you get rid of those extra ketone bodies if you're an uncontrolled diabetic. You either breathe them out in your breath, and the breath can actually have a kind of an acetone smell to it, and you also put it in the urine. All right. So if you measure ketone bodies, you don't have ketone bodies in the urine under normal circumstances, only during diabetes. Next, what about hemoglobin? So that means that I have disrupted a bunch of red blood cells releasing hemoglobin. So there's some sort of injury, maybe it's burn, trauma, whatever. Okay. Next, we have bile pigments. Now, bile pigments, normally we can have them, but normally we don't, but that can lead to there's some dysfunction in the liver or gallbladder. We can drop on down. What about erythrocytes? If I have red blood cells, that denotes some connection to blood. So that's usually an injury that is along the ureter, the bladder, or the urethra. All right. And the last thing is pus, white blood cells, immune cells. This denotes infection. That would be usually associated with the lower urinary tract. Okay. So this is not a complete list, nor is it meant to be, but it is an illustrative list of some of the things that you can use urine as a diagnostic. Okay. Last thing, and I'm done, and I'm almost done out of time too. I like to throw this one out here. How many of you have eaten asparagus and within a short time came, boy, my pee smells funny? Okay. Now, that doesn't happen to everybody. Only about 60% of the people can actually can smell it. It has not a function that they're not making that. It's in the asparagus, but depending on our sense of smell, may or may not have the ability to smell that. All right. I'm only familiar with this later study in the 2016, and basically found that 60% male and females basically could not smell. So I'm wrong. So only 40% of the population, if you can smell it, you're the minority. If you can smell it, you're the majority. Okay. So, and that doesn't matter. It doesn't have to be your own. It can be, you can be in a public toilet and you know someone who just, if you can smell it, you'll know. Okay. You can't hide it. All right. So with that, I'm out of time. I'm out of slides. We're done with the urinary system. I did make it despite the initial beginnings. We made it. So this is it before you wear. And so there is no quiz on Tuesday. That originally was in the calendar. I took it out. So what I want you to do, what I expect all of you to do is go wherever you're going later this week or not, enjoying whatever or not. And I'll see everybody on Tuesday for your final lecture. All right. And then we have our exam on Thursday. And then you're done. Okay. All assignments should be in. Right. Okay, everybody. Have a good end of the week, and I'll see everybody on Tuesday. Yep.